Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss the programmed cell death called oxieptosis. Oxieptosis, first reported in 2018, is associated with several diseases, including cancer, diabetic cardiomyopathy, osteoarthritis, lumbar disc degeneration, as well as vitiligo, where there are white skin patches due to the destruction of melanocytes. Now, one reason why we like to talk about this programmed cell death is because it's due to oxidative stress. Now, we talked a lot about oxidative stress in 8.8, video 8.8, and there are many diseases that are associated with oxidative stress. So this is an area that is extremely important for the healthcare workers. So when a cell experiences oxidative stress, one of the things it can do is die by oxieptosis. Now, there is a protein in the cell called KEAP1, Kells-like ECH-associated protein 1, or KEAP1. Now, this protein can sense and sort of quantify how much oxidative stress is there. So basically, the levels of reactive oxygen species can be sensed and quantified by this sensor protein. This is because KEAP1 has 27 cysteine residues. And remember, cysteine has the sulfhydryl group, and this sulfhydryl group is sensitive to oxidation. So by sensing the amount of oxidative stress, the amount of ROS, this protein can actually change shape and carry out its ROS sensing. Now, KEAP1 forms a complex with the transcription factor called nuclear factor erythroid 2 related factor 2, or NRF2, as well as the enzyme called phosphoglycerate mutase 5, or PGAM5. And this complex is formed close to the mitochondria, probably at the outer mitochondrial membrane. Now, when there is no oxidative stress, or when the oxidative stress is very low, so basically in a non-stress healthy cell, KEAP1 will have a certain shape. Now, this protein is part of the E3 ubiquitin ligase complex. So what this protein does, it will ubiquitinate NRF2 via K48 linkages. And remember, K48 link ubiquitin will cause the protein to undergo proteasomal degradation. So basically, in a non-stress cell, this NRF2 will be degraded and therefore present in very low concentration. Now, in a cell with moderate levels of ROS, what happens is the shape of KEAP will change as some of the cysteine residues get oxidized. So this change of KEAP1 shape is going to affect its function. Now it cannot mediate the ubiquitination of NRF2. So this NRF2 doesn't undergo proteasomal degradation. So now we have the NRF2 still alive and kicking in this cell that has moderate ROS. Now this NRF2 will be released and it will travel to the nucleus. Now, NRF2 is a transcription factor, so it will bind the antioxidant response elements of several genes within the promoter regions of several genes, and these are genes that code for antioxidant proteins. So these antioxidant proteins, examples include the enzyme NADPH quinone oxidoreductase, the antioxidant protein thioredoxin, as well as the antioxidant protein glutathione peroxidase. So increased expression of these genes is going to result in the increased amount of these antioxidant proteins, And these antioxidant proteins are going to help the cell in neutralizing these reactive oxygen species that it is facing. Now, in a cell with very high levels of ROS, now the keep is even more oxidized. Its cysteine residues more and more gets oxidized. It changes shape more drastically, and now it releases this PGAM5. So PGAM5 is now released from this keep 1. And this PGAM5 will now enter the mitochondria. In the mitochondria, the PGAM5 acts as a phosphatase enzyme, and it will remove the phosphate at a certain serine residue on the protein AIF, apoptosis inducing factor, which is located in the mitochondria. Recall that in the previous video on parthenatose, we talked about AIF. It's a mitochondrial flavor protein that was involved in parthenatose, which is a caspase-independent cell death. Now, in parthenatose, recall that the AIF went to the nucleus and degraded the DNA. But in oxieptosis, how this AIF causes cell death, that is still not clear. So that's an area of ongoing research. So to summarize, oxieptosis is a non-inflammatory programmed cell death. The membrane is intact, so nothing is released, so there's no inflammation. It is caspase independent, doesn't need any caspases, unlike apoptosis. The cell morphology of oxieptosis, it shares many similar features with apoptosis. It's also intriguing why a cell that is facing oxidative stress, why it would choose a certain programmed cell death over another. So there are several ways a cell can die by oxidative stress, including oxieptosis, apoptosis, necroptosis, as well as ferroptosis. Why one pathway over the other, we are still uncertain. So that's a little bit about this programmed cell death called oxieptosis. Once again, thank you very much for watching and supporting this channel. Until the next video, have a wonderful day.